Hey everyone, Dr. D here, and in this video we are covering Chapter 18 from our Talaro's Foundations in Microbiology 12th edition textbook by Barry Chess. So let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, Chapter 18. This chapter covers the gram-positive and the gram-negative cocci of medical importance. So let's go ahead and get started with some gram-positives. Let's talk about the Staphylococci. We're talking about the genus Staphylococci, not the shape and arrangement. I know this is very confusing for students. They're like, wait, I thought Staphylococci meant clusters of spherical cells. That is perfectly true. However, Staphylococci is also a genus, or Staphylococcus is a genus. Does that make sense? So technically it should be italicized here. In fact, I'm going to go ahead and italicize it. I think the publisher should have italicized it because they're not talking about they're not talking about the shape and arrangement. They're talking about the actual genus Staphylococci, right? So Staphylococcus is a common inhabitant of your skin and mucous membranes. These are spherical cells arranged in irregular clusters. That's what staphylo means. And then coccus means spherical cells. Staphylo means irregular clusters or grape-like clusters. These are gram-positive. They lack endospores and they lack flagella, so they are atrichous. They may or may not have capsules, and there are about 40 species within the genus Staphylococcus. And this is what Staphylococcus would look like under the microscope. Notice again, grape-like clusters of spherical cells. And so let's talk about Staphylococcus aureus. On a plate, on an agar plate, these grow in large, round, opaque colonies. The best incubator to use is the 37-degree incubator, because what does 37 mean? Can you beat Wicket? What does 37 degrees mimic? If you place a plate in 37 degrees, what is equivalent to 37 degrees? Why would that be the optimum temperature? Can you beat Wicket? That's right, Wicket. 37 degrees C is your body temperature. And so that's why one of the incubators in the lab is set to 37 degrees C. It mimics body temperature. Because Staphylococcus aureus grows on the skin, your body's temperature is probably going to be the optimum temperature for it to grow. Isn't that neat? And by the way, these colonies are usually yellow, because aureus, the first two letters of aureus are AU, and AU is the symbol for gold. These Staphylococcus aureus form golden colonies on a plate. Isn't that neat? They are also facultative anaerobes, which means they can grow in the presence or absence of oxygen. They can withstand high salt. That's why they can grow on your skin. Remember, your skin is salty, so they can withstand high salt. They are halophiles. They also can withstand pH differences. Remember, your skin is also somewhat acidic, so they can withstand that, and they can withstand somewhat high temperatures. And one thing about Staphylococcus aureus is that it produces many virulence factors. So let me tell you what a virulence factor is. A virulence factor is anything, you make a note of this, a virulence factor is anything that can give a pathogen the upper hand and help it be a better pathogen. So almost anything can be a virulence factor as long as it helps a microbe to cause disease. Does that make sense? Anything from fimbriae for adhesion to toxin production, all of those things are virulence factors. Here you can see those Staphylococcus aureus colonies on a plate. Notice that they grow with a golden pigmentation, And in this plate, you can see that they are growing on a blood agar plate. And you can see that they are breaking open those blood cells, right? That's called beta hemolysis. So you can see that Staph aureus is capable of causing beta hemolysis. That means complete lysis of blood cells on a blood agar plate. And that's because Staph aureus has many hemolysins, enzymes that break apart red blood cells. Look at this. They possess several different hemolysins. And these hemolysins are capable of lysing red blood cells. And yes, in case you're wondering, hemolysins, these enzymes are virulence factors. In fact, S. aureus is known for the ridiculous number of virulence factors it possesses. Everything on this table is a virulence factor of S. aureus. Coagulase, hyaluronidase, staphylokinase, lipase, penicillinase. I mean, the list goes on and on and on. Each one of these factors that S. aureus possesses helps it to cause disease. This is why when you do get a staph infection, usually they're referring to Staph aureus, the genus Staphylococcus, and the species aureus. It's a pretty bad skin infection, and it's difficult to treat. Why? Because look at the sheer number of virulence factors that it possesses. Now let's talk about the epidemiology and pathogenesis of S. aureus. Recall that epidemiology refers to the spread of disease, the study of the spread of the disease, and pathogenesis refers to how it causes disease. So we're talking about that. Staph aureus, for short, is present in most environments frequented by humans. It's readily isolated from fomites. I want to tell you what a fomite is. A fomite is something that's an inanimate object in your surroundings. A doorknob is a fomite. A lot of bodybuilders and gym rats pick up staph infections from the gym equipment. If you're in the gym lifting weights and someone had a staph infection or has a lot of staph on their skin and they use the workout machine and then you use it afterwards, that could potentially be a fomite where you picked up Staph aureus for your infection. That's why it's always good to wipe down that equipment before you use it. The carriage rate in a healthy adult is 20 to 60%. That means 20 to 60% of us healthy adults carry it around. It's part of our normal microbiota of our skin. The carriage is mostly restricted to the anterior nares. That's your nose, your nose holes, your skin, the nasopharynx, that's basically your throat area, and your intestine. And if you're predisposed to the disease, this is due to usually poor hygiene, poor nutrition, tissue injury, so you have a cut or something, pre-existing primary infection, diabetes, immunodeficiency. All of these can increase your chances of getting a staph infection. And unfortunately, although most staph nowadays is resistant to penicillin, there's a newer strain, not new, but newer strain that's even resistant to methicillin. And that's a dangerous strain of Staph aureus known as MRSA, which stands for methicillin-resistant Staph aureus. MRSA and its close other strain, VRSA, which stands for vancomycin-resistant Staph aureus, are very difficult to treat because they're so resistant to chemotherapies like antibiotics. Here you can see a lesion. This is known as a cutaneous lesion. That means a lesion on the skin, and that's a telltale sign of staph infection. Though these skin abscesses, basically staph causes a skin abscess, and it can vary in size, depth, and degree of tissue involvement. So this image shows you a somewhat minor abscess from Staph aureus, and this image shows you a much more severe abscess from Staph aureus. But both of these are Staph aureus infections. So like I said, these infections range from localized to systemic. Localized meaning at a particular spot on the skin, to systemic meaning throughout your body, through the blood, all over the body. Localized cutaneous infections, here's a list of them. These invade the skin usually through a wound, a follicle, or a gland. You can have folliculitis and hydradentitis. This is a superficial inflammation of the hair follicles and glands, respectively. These are usually resolved with no complications, but can progress. You could also have furuncles. Furuncles are boils. This is inflammation of hair follicle or sebaceous gland, which can progress into an abscess or a pustule. You could also have carbuncles. Carbuncles are larger and deeper lesions created by aggregation and interconnection of a cluster of furuncles. And you could also have impetigo. Impetigo are bubble-like swellings that can break and peel away, and they're most common in newborns. And again, all of these are examples of localized cutaneous infections due to staph. An example of a systemic disease would be if these staph got into the blood. That's known as bacteremia. That means bacteria in the blood, where the primary origin is bacteria from another infected site or medical device, and then it gets into the bloodstream and can even affect the heart. And these staph infections can be toxic. For instance, if you ingest these Staphylococci, that could lead to food intoxication. That means like essentially food poisoning and GI distress. Another result from these staph infections could be staphylococcal scalded skin syndrome, where the toxin from staph induces bright red flush, blisters, and then desquamation of the epidermis. That means your skin sloughs off. And it's called scalded skin syndrome because it looks like the skin has been burned and the skin is peeling off, but it's not because of a burn. It's because of this bacterial infection that you have the desquamation. Next, toxic shock syndrome. This is when you have toxemia. This means Staphylococcus toxins in your blood, right? Toxemia means toxins in the blood due to the bacterial infection, and this can lead to shock and organ failure. That's what toxic shock refers to. On the left, you can see scalded skin syndrome, where skin is sloughing off. It has an exfoliative feature to it, skin peeling away. And on the right, you can see bullous impetigo, which is characterized by bubble-like epidermal swellings that can break away and peel away. All of these are more advanced forms of staph infections. So here are some other members of the genus Staphylococcus to be aware of because they are important medically. Staphylococcus epidermidis, which lives on the skin. In fact, that's how it got its name, epidermidis, after the epidermis of your skin. This lives on the skin and mucous membranes. It can result in endocarditis, if it causes an infection, bacteremia, which means bacteria in the blood, or even UTIs, which are urinary tract infections. Staphylococcus hominis, this is another important species to know about. It lives around the apocrine sweat glands. These are the sweat glands near the armpits and the groin. Staphylococcus capitis, this lives on the scalp, face, and external ear. And all three of these may cause wound infections by penetrating through broken skin. Again, they're living on your skin. They're part of your normal skin flora, and they are commensal, which means they don't benefit you or harm you, but they benefit by living on you. But they are somewhat opportunistic pathogens, which means if you give them a chance, they could potentially cause disease. So let's say you get a cut in your skin, but you don't wash it out and try to apply an antiseptic on that skin, well, then that could potentially lead to a disease. Next, Staphylococcus saprophyticus. infrequently lives on skin, intestine, vagina, and can cause UTIs. And you might be wondering, how do clinicians diagnose staphylococcal diseases? Well, you would take a sample from either pus, tissue exudate, sputum, urine, or blood from the patient. You would then cultivate it. You can do a catalase test, biochemical testing, coagulase test, and you have these strips. These are called API strips, and these are the staph API strips. You inoculate each one of these little wells with the bacterial sample, a broth of bacteria. You incubate, and based on the colors that result, you can compare that to a chart which will identify the bacterium. Staphylococci are known for producing the enzyme catalase, which breaks down hydrogen peroxide. So for instance, this is Staphylococcus aureus, remember, with the golden pigmentation? And look, when you add hydrogen peroxide, and look at this, because they have the catalase enzyme breaks down the hydrogen peroxide, releasing oxygen. And that causes bubbling, right? The bubbles are due to oxygen released, and that's indicative of an enzyme possessing catalase. Notice that the bacterial species on the right is catalase negative because you added hydrogen peroxide, but you don't see that bubbling, indicating the absence of catalase. This one is a coagulase test where 5% rabbit plasma is inside of a test tube. You add the bacterium, and if the bacterium can produce the enzyme coagulase, it causes the rabbit plasma to coagulate or clump up, almost like a mucousy sample. And if the bacterium does not possess the enzyme coagulase, well, then the rabbit plasma does not clump up. There are also serological tests. That means tests that are based on antibody-antigen binding, and these tests are used in the clinic as well to indicate the presence of Staphylococcus. So here you can see you have staph. If you have agglutination, that means clumping up of these microbeads with antibody. And here you can see this would be a negative for staph because you do not have that agglutination or clumping up with the microbeads with antibody. All of these, by the way, all of these are tests that are done to identify staph. So if you want to identify staph in a patient, you could do the API test, you could do a catalase test, coagulase test, and these serological tests. All of these are common tests to confirm the presence of a staph infection in a patient. Now let's discuss some clinical concerns regarding staph infections. Did you know that 95% of Staph aureus possesses the gene penicillinase? Do you recall what penicillinase does? We talked about it before. Can you beat Wicket? That's right, Wicket. Penicillinase is an enzyme that bacteria use to chop up penicillin, right? To attack the beta-lactam ring of penicillin and render it useless. So let me ask you this. If 95% of Staph aureus have penicillinase, then do you think penicillin is readily prescribed for staph infections? Would that be logical to do? That's right. That's right, Wicket. No, staph pretty much are almost 100% resistant to penicillin nowadays. So instead, we use methicillin. Unfortunately, like I said before, now there are methicillin-resistant Staph aureus as well, which carry multiple resistances. This means that they are resistant to a host of different antibiotics, and they can be very, very tricky to resolve and to treat. Some strains have resistance to all major drug groups except vancomycin. But like I mentioned, there's even a new strain of staph known as VRSA, vancomycin-resistant Staph aureus. So effectively, yes, there are strains of staph that are essentially resistant to all antibiotics, which is very scary because at that point, all you can do is just remove tissue or remove limbs in order to resolve the infection. This is known as a post-antibiotic world, right? Post-antimicrobial world where, where the microbes have adapted to the point where they are now resistant to antibiotics. And that is a terrifying thought. So how do we treat staph infections? Well, you have to select the correct drug. It depends on the sensitivity testing. Remember the Kirby-Bauer test that we talked about previously in a previous chapter? A Kirby-Bauer test must be performed to determine which drugs Staph aureus would be susceptible to. The less resistant forms of Staph aureus can generally be treated with cephalexin, sulfa drugs, tetracyclines, and clindamycin. MRSA, on the other hand, there are still a few drugs that will generally work, including vancomycin, ceftaroline, linezolid, and daptomycin. However, like I said, there are new strains of staph, including VRSA and VISA, and these are essentially resistant to everything, though quinupirstrin and dalfopirstrin are the drugs of choice. All right, they're the last bastion that we have to reach for to try to treat these VRSA and VISA infections, which are highly resistant forms of staph. And because staph infections can be so devastating, universal precautions by healthcare providers to prevent nosocomial infections are in place. I want to define this term nosocomial for you. It's an important term you should know. Nosocomial refers to diseases that are picked up at a clinic, right? So in a hospital setting. Let's say you go in because you have an infection in the foot, but then while you are there, you pick up a staph infection somewhere else, or you pick up a C. diff infection while you are at the hospital. That would be known as a nosocomial infection. And so Staphylococcus aureus is a typical or common nosocomial infection. It's something you pick up at the hospital, but it's not the reason you visited the hospital. So hospital workers who carry S. aureus nasally may actually be barred from nurseries and operating rooms and delivery rooms because of the risk of spreading that Staph aureus to vulnerable patients. And hygiene and cleansing are very important if you have staph, making sure that the patients are not exposed to Staph aureus during their treatments. Okay, so that leads us to concept check number 1. Which test differentiates S. aureus from other staphylococcal species? Is it the Gram stain, the catalase test, oxidase test, or coagulase test? Now, I didn't mention this. I should have mentioned this. Let's go back real quick. I want to show you something. I should have mentioned this, but Staph aureus is coagulase positive, which means it produces the enzyme coagulase, which causes rabbit plasma to coagulate, whereas the rest of these staph species are coagulase negative. Does that make sense? So I'm sorry, I should have mentioned that earlier. The answer would be coagulase, and that's because the other Staphylococci species are coagulase negative, while Staph aureus is coagulase positive. And by the way, this is another important point for you to write down. Staphylococcus is known for being catalase positive, okay? Make a note of that. Staphylococcus is known for being catalase positive, while our next character, Streptococcus, the genus Streptococcus, is catalase negative. You should write down these are catalase negative. Staphylococcus are catalase positive, Streptococcus catalase negative. So these Streptococci are gram-positive spherical to ovid cocci arranged in long chains. That's what strepto means. Strepto means chains, and cocci means spherical to ovid cell shape. They can commonly appear in pairs, but also long chains. They are not endospore forming. They are non-motile, so they are atrichous. Some can form capsules and slime layers, others don't. They are facultative anaerobes. That means that they can either live in the presence or absence of oxygen. They do not form catalase. Remember, they are catalase negative. However, they have a peroxidase system, so they are peroxidase positive. Most parasitic forms are fastidious. Do you guys remember what fastidious means? I like to think of it as finicky. Okay, fastidious bacterium are bacterium that will not grow unless they are given enriched media. So, for instance, blood agar, where blood cells enrich the media. They are small, non-pigmented colonies, sensitive to drying, heating, and disinfectants. Again, this is what Streptococcus looks like. You've got chains of spherical to ovid cells. And these Streptococci, these species are grouped based on what are called Lancefield groups. This refers to the carbohydrates they have in their cell wall. There are 20 groups, A, B, C, etc. You can also classify these Streptococci based on their homolysis, right? Do they do alpha homolysis, beta homolysis, gamma homolysis? What type of homolysis do they do? Though most of them do a form of homolysis. Look here, groups A, B, C, G, and some D strains are beta hemolytic. Remember, they can completely lyse and completely break open red blood cells, whereas S. pneumoniae, Streptococcus pneumoniae, and Streptococcus viridans, these are alpha hemolytic, right? They are able to partially rupture those red blood cells. So again, you can see here the separation of the streptococcal subgroups. If you want to figure out which streptococcal species you're talking about, if it's beta hemolytic, remember you have this clear area, colorless halo surrounding the colonies. That means that you need to do another test, a bacitracin test. This is an antibiotic. If the bacteria are beta hemolytic and bacitracin sensitive, then it's a group A streptococci. For instance, Staphylococcus pyogenes, the bacteria that causes strep throat. If it's bacitracin resistant, then it belongs to group B or C streptococci. Now, if it's alpha hemolysis we're talking about, that means the bacteria partially ruptures the red blood cells on a blood agar plate. Then we must do another antibiotic test, optogen. If it's optogen sensitive, it's Streptococcus pneumoniae. If it's optogen resistance, then it's a group D and Viridans streptococci. So again, this is kind of the flow chart for understanding or identifying strep species, I should say. You would do a catalase test, and let me see if you could beat Wicket. Should it be catalase positive or catalase negative? That's right, Wicket. Streptococci are catalase negative, gram positives. If it's beta hemolytic, you would also do a bacitracin test. If it's alpha hemolytic, you would do an opsinin test, and you would test to see which species you have. That way, that's the way you would identify the strep species in a lab. Now, the most serious streptococcal pathogen is known as Streptococcus pyogenes. It's a strict parasite, which means that it's looking to cause disease. It feeds off of your tissues, right? Parasites become nutritionally dependent on a host. And this is the bacterium that's known for causing strep throat. If you've ever heard of strep throat, that's what we're talking about, Streptococcus pyogenes. It inhabits the throat, nasopharynx, or occasionally the skin. These are some of the virulence factors that S. pyogenes possesses. And remember, virulence factors help a microbe to cause disease. And here are some extracellular toxins that it possesses. You see, these are the toxins it possesses, and enzymes, extracellular enzymes or exoenzymes it possesses that cause damage as well. Look at all these toxins and enzymes that it secretes that can cause damage to your tissues. And humans are the only significant reservoir. That means that you can find this species on humans, and that's where it normally resides, in a human throat. Transmission involves contact, let's say kissing or contact, droplets from, let's say you're spraying it instead of saying it, food, sharing food, touching things after you've touched your mouth, etc. And these can lead to skin infections, right? Some of the more mild infections include impetigo, which is, again, a superficial lesion that break and form highly contagious crust, often in epidemics in school children. You could see here, this is impetigo. It forms a yellow crust that is highly contagious. It can also form erysipelas, which is a pathogen which is when the pathogen enters through broken skin, spreads to the dermis, in the skin, and subcutaneous tissues. This can be superficial or even go systemic. And here you can see when it's breaking through the skin and going down into those sub tissues. What's worse is that Streptococcus pyogenes can also cause what is known as necrotizing fasciitis, also known as flesh-eating disease. This is when it enters from cuts or abrasions on the skin, spreads rapidly through the subcutaneous tissue, leading to severe disfigurement or even death if not treated. The problem is enzymes digest connective tissue while toxins poison the epidermal and dermal tissues. Some toxins can act as superantigens, which means that they trigger your immune system to overreact, and that causes even more damage. Here you can see necrotizing fasciitis eating through, a person's leg. It's a devastating infection. And by the way, these streptococcal diseases are distinguished based on the depth at which they occur. So for instance, look at this. This is your skin layer right here. This is an anatomical sketch of a skin layer, or your skin layers, I should say. If it's up here, it's called the epidermidis. And if the strep affects the epidermidis, this is what leads to impetigo, erysipelas, and ecthyma. These diseases occur near that upper epidermidis. However, cellulitis occurs way down here in the superficial fascia. Necrotizing fasciitis occurs all the way down here in the subcutaneous fat, nerve, arteries, veins, in the deep fascia. And myonecrosis occurs deep down into the muscle area. So this is where they get their names. It's like, where did the Streptococcus go to cause infection? And that's going to give you the resultant infection. Again, Streptococcus pyogenes causes strep throat, also known as streptococcal pharyngitis. The pharynx and the tonsils, see, this is the back of the throat where the tonsils are. The pharynx and the tonsils become bright red and suppurative. The white areas are pus-containing nodules. And in severe cases where the tonsils meet, they are said to be kissing. You see, so the tonsils are so swollen. Look, the tonsils are so swollen that in severe cases, the tonsils can actually touch. And that's known as kissing. By the way, this bacterium is called Streptococcus pyogenes. And pyo refers to pus. That's how it got its name. These pustules that form in the back of the throat are characteristic of a strep infection, a Streptococcus pyogenes infection, and pyo means pus. So now you know, pyo refers to pustules. And strep can cause systemic infections as well, like scarlet fever, septicemia, pneumonia, and streptococcal toxic shock syndrome. Long-term complications of group A infections include rheumatic fever, which follows overt or subclinical pharyngitis in children. Carditis with extensive valve damage is possible, as well as arthritis, chorea, and fever. In fact, let me show you here. This is a complication with rheumatic fever, and notice that it's destroyed a patient's heart valve, and this patient needs a pig heart valve replacement due to this bacterial systemic infection. And again, another long-term complication of group A infections is acute glomerulonephritis, which is a type of nephritis. This is where your kidneys become inflamed and it can lead to kidney failure. And that leads to concept check number 3. Which of the following is not a possible complication of strep throat? Rheumatic fever, yes, we just talked about that. Scarlet fever, yes. Acute glomerulonephritis, yes. So the answer has to be otitis media. Otitis media. And by the way, with that, let's go ahead and take our break time with Gizmo and Wicket, see what these little guys are up to, and we'll be right back to finish off this interesting chapter. All right, welcome back from break time with Gizmo and Wicket. So we just finished talking about Group A streptococcal diseases. Now let's talk about Group B, starting with Streptococcus agalacetae. These are regularly reside in the human vagina, pharynx, and the large intestine. They can be transferred to the infant during delivery and cause severe infection this way. In fact, it's the most prevalent cause of neonatal pneumonia, sepsis, and meningitis, right? So during birth. Pregnant women need to be screened and treated in order to prevent this meningitis or pneumonia from spreading to the child during the birthing process. What about Group D streptococci? Enterococcus faecalis, Enterococcus faecium, Enterococcus durans. These are normal colonists of the human large intestine, though they are opportunists, which means they can cause disease, urinary, wound, skin infections, particularly in debilitated persons. And then groups C and G, these are common animal flora. They live on our bodies, frequently isolated from our upper respiratory tract. They can cause pharyngitis, glomerulonephritis, and bacteremia. Okay, and then there are the alpha hemolytic streptococci, also known as the Viridans group. It's a large group including Streptococcus mutans, which, by the way, this is the one that causes a lot of dental caries, which means cavities on our teeth. Streptococcus oralis, Streptococcus salivarius, Streptococcus sanguinis, Streptococcus milleri, and Streptococcus mitis. Many of these are found in our mouth. Most numerous and widespread residents of the gums and teeth, oral cavity, and also found in our nasopharynx, genital tract, and skin. And the Viridans group can cause bacteremia if it goes into our blood, meningitis, abdominal infections, and even tooth abscesses. However, the most serious infection by this group are subacute endocarditis, which is when the blood-borne or bacteremia, the blood-borne bacteria settle and grow on heart linings or valves, causing heart damage. Here you can see that heart damage. The streptococci have colonized parts of the heart. Bacterial vegetations can be seen along the upper edge of the surgically open mitral valve. The vegetations, which are the white patches there that you can see, constantly release bacteria into circulation, and this is particularly bad. And as I mentioned before, Streptococcus mutans, really neat. This is the bacterium most associated with adhering to your teeth, causing plaque, right? Your plaque, that the reason you have to brush your teeth. They're involved in dental caries, which is a fancier way of saying dental cavities. Next, let's talk about Streptococcus pneumoniae. This is a bacterium that causes 60 to 70% of all bacterial pneumonias. Pneumonia is inflammation and fluid in the lung. These small lancet-shaped cells arranged in pairs and in short chains, they're the ones that are responsible for the pneumonia. And all of the pathogenic strains of the pneumoniae, they form large capsules, which is their major virulence factor, right? Because encapsulated bacterium are very difficult for your immune system to treat and clear. And again, these cause pneumonia and otitis media, which is an infection of the middle ear. Here you can see the bacterial pneumonia at work, Streptococcus pneumoniae in the lungs. As the pneumococcus traces a pathway down the respiratory tree, down your trachea, the bronchus, and the bronchiole, it provokes intense inflammation and exudate formation. Exudate is fluid that leaks out of blood vessels. The blocking of the bronchioles and alveoli are these little sacs that are at the ends of our bronchioles and responsible for gas exchange, oxygen, and CO2 exchange. Well, they become inflamed and full of fluid. And on the right, you can see otitis media. Remember I said that's a middle ear infection where the bacterium makes its way up the eustachian tube from the upper respiratory secretions to the middle ear, causing an ear infection. And that middle ear infection is very common among children, otitis media. On the left, you can see that acute inflammation and internal pressure of the middle ear. You see how the middle ear is red and it's kind of bulging. That bulging is showing you the tympanum as seen during an otoscopic examination. So you're looking inside of the ear and you see this redness and bulging. And in children with chronic infections like this, it may be important to surgically insert a tube inside of the tympanum to equalize the pressure of the middle ear and the outside, so minimizing the pain and the pressure involved here. So how do we treat these pneumococcal infections? Usually with penicillin G or V. Though due to increased drug resistance, we've had to change tactics. Several vaccines are available for high-risk individuals. All right, and that leads to concept check. Number 5, bacterial pneumonia by Streptococcus pneumoniae is highly contagious. And actually, the answer here is false, and I'll tell you why. Let me go back to here. See, high-risk individuals are people, older adults and children, okay, and young children under 2 years old. If you have a robust immune system, then it's actually more rare to get this pneumococcal infection. So that about wraps up the gram-positive cocci that I wanted to get to. Now we're going to talk about the gram-negative cocci. So everything else in this chapter are the gram-negative cocci that are of medical importance. Let's start with the family Neisseriaceae. These are residents of mucous membranes of warm-blooded animals like you and me. The genre that we're going to talk about are Neisseria. The main one's Neisseria. 2 primary human pathogens. Neisseria gonorrhoeae, which causes gonorrhea, and Neisseria meningitidis, which causes meningitis. So we're going to talk about those 2 in particular. Again, these are gram-negative bean-shaped diplococci. That means they hang out in pairs, though they aren't perfect circles, right? They're not perfect cocci shape. They're bean-shaped. They do not develop flagella nor endospores. They do have capsules, right? The pathogenic strains are capsulated. They have pili. They are strict parasites, which means that they become nutritionally dependent on hosts. And did you know that they do not even survive long outside of the host? They require a host in order to live. They are aerobic or microaerophilic. They produce catalase and are cytochrome oxidase positive. So here you can see that genus I'm talking about. Look at this. It looks like little hearts. Well, those aren't little hearts. This is a scanning electron micrograph of Neisseria gonorrhoeae. Notice that you can see the bean shape. This is Diplococcus. See, here's one bean-shaped cell, and next to it is the other bean-shaped cell, and the flat side of the bean-shaped cells are what's sticking together. So these are diplococci technically. Now, let's talk about Neisseria gonorrhoeae. This is the one that causes the STD, or sexually transmitted disease, gonorrhea. And these bacteria have virulence factors that lend to their pathogenicity. That means their ability to cause disease, including fimbriae. Remember, these are short bristle-like extensions that help the bacteria to attach to your tissues, to your cells. And they can slow phagocytosis by white blood cells. They also have IgA protease, which cleaves secretory IgA. Remember when I said that your saliva and your mucus has IgA inside of it? This is a type of antibody that helps to fight bacterial infections. And so these guys produce a protease that destroys this important antibody that you have inside of your secretions. What about the epidemiology? Remember, epidemiology is the study of spread of disease, and pathology, which is the study of the actual infection and disease of gonorrhea. It's a strictly human infection. It's in the top 5 of sexually transmitted diseases. This is an interesting fact. Do you guys know what the infectious dose refers to? That means how many bacteria does it take to make you sick, right? The infectious dose is between 100 and 1,000 colony-forming units. So it takes between an inoculum of between 100 and 1,000 bacterium to make you sick. And again, it does not survive for more than 1 or 2 hours on fomites. Remember, because it doesn't survive well outside of the body. Fomites are inanimate objects like doorknobs and things. So if you get Neisseria gonorrhoeae on your hands and you touch a doorknob, it will not last on that doorknob for long. Hi, look, you guys, it's Wicket. Wicket has come and graced us with his presence. Hi, Wicket. Are you here to answer questions for Okay. Wicket is here, you guys. That's great. I don't call it a cameo when he shows up. I call it a catmeo. Get it? Catmeo. I know it's cheesy, but you know, I do the dad jokes. I'm the source of the dad jokes over here. Guilty as charged. All right, here you can see gonorrhea over the years. It's what's known as an endemic, which means that it's a disease that just it's constant throughout the population throughout years. There's always some low level of it. And you can see in the 60s, the late 60s and early 70s, where there was a sexual revolution, you know, you can see that the numbers of this disease spiked, you know, and then, you know, with more education, it's tapered off to some extent. Now, what happens when a male contracts gonorrhea? This is known as urethritis, which is infection of the urethra, basically. The urethra is the passage, you know, that urine takes outside, you know, out of the body through the genitals. The urethra has a yellowish discharge, scarring, and can even lead to infertility. So, and this is what happens. By the way, 10% of males are actually asymptomatic for gonorrhea. 90% experience this yellow discharge through the penis. What happens is somewhere near the urethral opening, there is the infection begins, and then the infection travels up the urethra, down the vas deferens, and can even infect the testes themselves, causing scar tissue to form, and ultimately can result in infertility as well. In the female, this bacteria causes vaginitis, urethritis, salpingitis, essentially PID, which stands for pelvic inflammatory disease. Again, the bacteria makes its way up the cervix, through the uterus, can even make its way through the fallopian tubes and cause a lot of infection and scar tissue to form there. Again, it can also lead to infertility if not treated. If the mother has gonorrhea, then the child can even pick it up during birth. This can lead to eye inflammation, resulting in blindness in the child if not treated. You can see here what is called gonococcal ophthalmia in the newborn. So here you can see that eye infection in the newborn, the gonococcal infection passed on from the mother to the infant during birth. You see gonococcal ophthalmia neonatorum in an infant infected during birth. This can lead to blindness if not treated properly. And remember, these are gram-negative bacterium, so if you do a gram stain, these will be pink, right? They are gram-negative bacterium. Here you can see that gram stain. These have been gram stained, and you can see the tiny gonococcal here, these little diplococci, those diplococci, a bean-shaped bacterium. And these here are actually neutrophils trying to phagocytose them. Isn't that neat? So this is a sample taken by a patient with gonorrhea and stained. And you can see it shows positive for gonococcal infection. That's how they can diagnose the gonococcal infection, by taking, sample of the pus. Now let's talk about Neisseria meningitidis. This is the bacterium that causes several instances of meningitis. Here are a list of its virulence factors. You see, it has several virulence factors, which means that it can be a tough pathogen. Humans are the reservoir. The reservoir, again, reservoir refers to where do you normally find this particular pathogen? Where does the pathogen normally reside? That is what's known as the reservoir. And humans are reservoirs for this Neisseria meningitidis. You can find it in the nasopharynx, the upper respiratory throat area. 10% of the adult population carry it without symptoms. However, if this bacteria were to enter the bloodstream, cross the blood-brain barrier, permeate the meninges, which is the membrane surrounding the brain and the spinal cord, and grow in the cerebrospinal fluid, then it causes meningitis, which is inflammation of those meninges, the membranes surrounding your brain, surrounding your spinal cord, and this is particularly devastating. So here's how it works. Again, you normally have Neisseria meningitidis in the upper respiratory tract, in their nasal cavity, nasopharynx area. However, if it were somehow to pass from the sinuses into the brain cavity like this, you see, if it were to get into the brain area, the bacteria spread to the roof of the nasal cavity, which borders a highly vascular area at the base of the brain. From this location, they can enter the blood and escape into the cerebrospinal fluid, into right here. This is the cerebrospinal fluid. And then cause infection in the meninges, leading to meningitis, inflammation of the meninges. And this results in inflammatory, purulent exudate over the brain surface. And this swelling of the brain membranes can actually result in death. So this is highly, highly risky infection. And to make it worse, the release of endotoxins during meningococcemia, that means, these bacteria into the blood, may cause hemorrhage, coagulation, and vascular damage, leading to necrosis, that means death, of tissues, especially in the extremities. Crops of lesions called petechiae develop on the trunk and appendages in about half of the cases. Here you can see just how devastating this can be. One clinical sign of meningococcemia is the release of endotoxins during the meningococcemia, which can cause hemorrhage, coagulation, and vascular damage, leading to that necrosis, the death of tissue, especially in the extremities. You see here, this poor child has this disease in the extremities, in both legs, due to that meningococcemia, which means, the bacteria has made its way into the blood and gone systemic. This can be usually diagnosed by gram stain, CSF, blood, or nasopharyngeal sample. A rapid test is done for capsular polysaccharides. If the patient is positive for this bacterium, they're treated with type 4 penicillin G, cephalosporin as well, prophylactic treatment of family members to stop the spread, and anyone else who was in contact with this patient. And there's even a primary vaccine which can be administered as well ahead of time. Now, other important gram-negative cocci or coccobacilli to talk about, the genus Moraxella. Moraxella catarrhalis is found in the nasopharynx. It's a significant opportunist in cancer. So unfortunately, patients with cancer, diabetes, or alcoholism, this opportunist can cause infection. Another important gram-negative is the genus Acinetobacter. Acinetobacter baumannii targets wounds, lungs, the urinary tract, burns, and blood. It's a major nosocomial. Remember, it can spread in the hospital setting and community-acquired pathogen as well. So concept check number 7, a common cause of meningitis in young adults is found in the genus, is it Streptococcus, Staphylococcus, Neisseria, or Moraxella? Remember, it's Neisseria. Remember, Neisseria meningitidis. By the way, I just want to make something clear to you guys. Meningitis simply means inflammation of the meninges, the membrane surrounding the brain and the spinal cord. Many different pathogens can cause meningitis. So Neisseria meningitidis is a major cause of meningitis, but it's not the sole cause of meningitis. Just be aware of that. There's also viral meningitis, also with pneumonias too. Remember Streptococcus pneumoniae? It's a bacteria that causes pneumonia. Well, guess what? Pneumonia simply means inflammation or fluid in the lungs. Many different germs cause pneumonia. Streptococcus pneumoniae is a major cause of pneumonia, but it's not the sole cause of pneumonia. Though some diseases do have a sole cause. So do you remember we talked about gonorrhea? We talked about Neisseria gonorrhoeae. Well, that is the only and sole cause of gonorrhea. So what I'm trying to get across to you is that some diseases are caused by a host of different germs or pathogens, while other diseases have one specific cause. And with that, we have ended this chapter 18. I hope you learned a lot about the medically important coccus-shaped bacterium, both gram-positive and gram-negative. Please let me know if you have any questions, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.